waiting for the others or no? Yes, okay. Thank you for coming.
Thank you. Oh.
Okay. Now it's good. Shall I start now? I think we should start this afternoon session. Thank you for coming. Okay. Good afternoon for everybody. I think we should start this program. I would like to introduce you, Professor Susan Brain from King's College London. She will give a talk for you this afternoon, and you can see the title. The title will be, it's a very good story in the drug development, a story of drug discovery today about CGRP in migraine and beyond. Sue Brain is absolutely good expert in the field of neuropeptides. She published a basic paper in the Nature many years ago about the vasodilator effect of CGRP in humans. After that, she started to work with TRP channels, TRPV1 receptors and TRPA1 receptors, and published a lot of paper in this field. First time I met her exactly 1996 here in Pécs. We organized the meeting for the European Neuropeptide Club, and this was first time I met her, but later on I was invited to work in her lab, King's College London, as a young postdoc. I dealt with the NK1 receptor, the role of NK1 receptor, substance P and CGRP in pain and inflammation. And later on, she also invited my younger colleagues to stay and to work in her lab. For example, Gábor Posga is still in the audience. He worked with her almost 2 years, 2006 or 8 maybe, something like this. And therefore, we decided she would be an excellent tutor and very excellent supporter in this program. Sorry, I have to go there because the other audience from outside can see me. And now I would like to ask Sue Brain to start her talk about the role of CGRP in migraine and about the drug discovery process. Sue.
Thank you very much. Okay, so thank you. What I thought I'd try and do today is sort of take you through my career in a way, but also concentrate on the drug discovery story relating to CGRP. Because just like any career, there's lots of ups and downs, and I've been involved in most of them. So that's what I've decided to do. So moving on, when you think of the drug discovery and development timeline, you think of various different phases, and usually as an undergraduate you learn about these. You learn about the first phase when someone decides on a target, and then they realise that they need to start investigating for compounds. At that time, the chemist is heavily involved, and it's easy to generate lots of compounds. What you're looking for is just one sort of hit initially, and then you can look at all sorts of related compounds to work out which ones really do affect that enzyme or receptor, or stimulate if you're looking for an agonist. Then you'll get quite a few, perhaps a whole range from one class. You've decided on the class of compound you want, and you can test them out preclinically. And you'll look at the bottom, there's a number of years suggested, typically for how long that takes. It's certainly not too short. Then you'll move on to clinical trials. Here it's suggested you take 10 compounds into clinical trials. Others may say that's quite a few. But what you're after is one that you could either take to the European agencies or to the FDA in America and have a drug approved. If you get to this point and there aren't hardly any side effects, you're onto a winner. You're helping patients, you're helping your company, and you're helping your nation, etc., etc. So that's what's meant to happen. It's got stuck, I think. Let's see if I can press it on. Here we are. And the disease that we're going to sort of talk about, of course, is migraine and CGRP. But the point I wanted to point out that's right down the bottom here is that it can take a lot longer. And the story of CGRP started when it was discovered in 1982, and there's all these ups and downs that I'll tell you about. And there wasn't a drug licensed until 2018. So with respect to CGRP discovery, we had to learn about the biology as well and where CGRP is found. CGRP was discovered in the early 1980s. It was discovered through molecular biology. Erica mentioned substance P, with which it's colocalized. That was discovered from just realizing there was a substance there and purifying it. But CGRP was one of the first studies discovered by molecular biology. There's two forms. There's a alpha form and a beta form. Alpha is generally considered to be the major vascular phenotype, although now people have realized that there's crossover. And it's contained in and released from sensory nerves. And these are commonly known as the pain fibers, as I'm sure you know. So how did it get this name, CGRP, calcitonin gene-related peptide? Well, the calcitonin gene, which I'm sure most people here know, that's involved in regulation of bone metabolism, had been known for some time. And people were studying it, and they realized that under certain circumstances, you had an alternative splicing of the gene. So normally in bone, you get calcitonin produced, and that's a 32 amino acid peptide. But in sensory nerves, or when the discovery was made, it was realized in aging rats or in people with medullary thyroid carcinoma, you get CGRP produced. It wasn't really realized that it was found in sensory nerves until people made an antibody to it and looked to see where the antibody bound. So this was one of our first experiments where we injected a certain amount of this peptide into human skin. We weren't quite sure quite how much, but it had a very different effect to histamine. We injected histamine, got a typical triple response, and that disappeared after a few minutes, that response you get when you're stung by a nettle or something like that. With CGRP, we definitely got darkening of the skin due to increased blood flow. And in this first picture, it was because we injected quite a lot, I think, we got these little sort of legs appearing. They look like legs of some sort of sea urchin. And what we think is that CGRPs remained not broken down, and it's got into the lymphatics. So, and there it's innervating the small blood vessels around it. We did rather more considered pharmacological experiments, I think you might say. We did a dose-response curve, and you can see here CGRP at even very low amounts causes what I'm hoping you can see is a local reddening, and that's due to increased blood flow. Substance P, by comparison, causes this triple response. You get a bit of swelling, and you get a axon reflex flare. So it can kick off that, possibly by releasing histamine, but it's not just a typical straight vasodilator in skin. Go down to lower levels and you can't see much. CGRP acts on its receptor, and the receptor is G protein linked. It's called CLR, calcitonin-like receptor, and that's associated with a single transmembrane component, RAMP1. This is quite rare to have these two components there. It took a long time from when CGRP was discovered before this was realized. But then, since it's realized that this is the receptor, through cyclic AMP leads to increased blood flow and vasodilatation. And indeed, CGRP is a very potent microvascular vasodilator. And if you give enough to a human, you will get hypertension. Lars Edvardsen and Peter Goseby and one or two other people who studied migraine realized there was a lot in the cerebral vasculature, and they started to look for evidence in migraine. Now, you probably realize that migraine is certainly something slightly more severe than a headache, but it accounts for quite a lot of headaches. And this is a typical picture, I think, because you can see that in the side where there's the migraine and the pain, there's involvement from the trigeminal nerve sort of endings that innervate the eye, and this is quite common. There's a huge amount of art that you can view, and the Wellcome Collection in London's tried to collect them together, that's been drawn by migraineurs to show what they see during an early phase that's called the aura. And you can have migraine with aura, where you get very disrupted vision. The positive side is that it can tell you you're going to get a migraine, and you can take a tablet potentially during this phase. But the negative side is that it just tells you you're going to get a migraine. So there's a high global prevalence, and people suffer ill health. It can come and go as you go through life, but people suffer ill health. They get attacks of severe headache, pain on one side, like I said, increased phonophobia and photophobia, sensitivity to light and sound. There's gut side effects. There's a predominance in females, and it's associated with the menstrual cycle, so hormones are involved as well. Migraine without aura is commonly called a common migraine. A migraine with aura is classic migraine. The financial impact is absolutely huge. So it's not just an impact of personal burden. In the UK, where we have our National Health Service, and you can quantify these things quite easily, it costs up to £1 billion per year. And if you think of the economic costs, because people commonly take days off with migraines, it will be a lot worse. So, Don't know what's happened there. But that should say, where is the pain? And the dura is, or the meninges, the brain layers, are sensitive except for a few areas. And within there will be the dura mater, which is considered to be one of the pain-sensitive areas. And we also have the arachnoid and the pia mater as well. So the meninges are very densely innervated with sensory nerves. Animals you may want to study preclinically are similar. And they receive blood from the middle cerebral pial and middle meninges arteries. And in humans, experimental stimulation of these vessels is known to lead to migraine pain, and this involves the trigeminal-vascular system. So this is from AstraZeneca, when we were starting to think about where nerves and blood vessels fitted in. And they tried to discuss of the blood vessels, basically the cranial blood vessels innervating the pain. And then they talked about the trigeminal vascular system. This could well be just outside the blood-brain barrier, but then when you get to here, you're usually inside the blood-brain barrier. So there's areas that will be beyond the blood-brain barrier. So in addition, you have stimulus of trigeminal nucleus caudalis, and then this can innervate upwards and backwards and accelerate this pain, and use through the dorsiraphe nucleus and the locus coeruleus give us some indication of the gut pain. So quite a lot is known about the anatomy, but the pain itself is not completely understood. Certainly, we know that migraine involves dysfunction of the brain stem that normally modulates the sensory input. And we know, just like I tried to say, there's the three key factors. There's the cranial blood vessels in the dura, there's the trigeminal nerve innervation of these blood vessels, there's trigeminal links with brainstem that, of course, transmit and modulates information to the cortex via autonomic reflexes back to the dura, etc. So, where are we with the drugs and treatments, and why do we want new drugs? So, there's two approaches. We've just come from a conference where migraine treatment has been discussed, and there's a preventive approach which is suitable if you suffer quite a few migraines. And people are now suggesting that preventive medicines should be given perhaps at an even more earlier stage than I've got here, because it is hoped that they keep everything in a stable situation in the brain. And then there's reactive treatments as well, and they're taken to combat and treat the attacks. And generally, patients, from my experience, seem to be happier to think about reactive treatments. Some of these drugs you will know from day one of medical school, really. There's the non-specific treatments, such as non-steroidal anti-inflammatory drugs, aspirin, naproxen, and they're used as reactive treatments. The first specific treatment for migraine was the triptans. And there's also wherever you go, you'll find there's herbal conditions. This is one that's considered popular in the UK, but there won't be any double-blind clinical trials to show it's better. So the triptans were very useful for us in learning more about CGRP and migraine. Serotonin, 5-HT, sort of is involved in mood very much, and generally thought to be involved in good mood. And there's 5-HT receptors, up to loads of them, up to about 7, And the 5-HT1B1D receptor agonists were investigated in the 1980s. And it was realized that a drug developed from a rye fungus and developed chemically, some people talk about these drugs, the triptans being called the grandchildren from the rye fungus, have very simple pharmacokinetics, and they have efficacy in migraine based on well-designed clinical trials. They're not effective in tension headaches, the sort of headache you get from too many lectures in a day, etc. But they do have moderate side effects. They're actually vasoconstrictors in coronary arteries, so there's restrictions on their use in cardiovascular disease. Also, they'll cure the migraine in the patients that respond to them, but then the next day you'll find that there's a rebound migraine. So patients would say, I want something that's better. So it was realized that as you have a migraine, if you measure the drain-in jugular vein, that you can measure raised levels of CGRP. Some people, Peter Goldsby and Lars Edvardsen, that I mentioned before, realized this. They also realized that if you took triptans, that as the migraine got better, the level of CGRP they could measure in the drain-in jugular vein was reduced. So we know the nerves that innervate the dura, where it's thought the pain all originates from, can release CGRP, and this is a blow-up. The CGRP, when released, can vasodilate arteries, and it's thought that this may contribute to some of the pain. CGRP is released, and it will act on the smooth muscle or endothelial cell receptors to mediate vascular relaxation. Triptans, the 5-HT1D1C receptor agonists, can inhibit the release of CGRP. It's also thought they may inhibit some of the increased blood flow that's caused by CGRP. So whatever happens, the big link with migraine was that the triptans can make it better, and they can reduce the link with CGRP. So they did some experiments that were never really repeated, and have never really repeated until quite recently, that you can measure CGRP in the drain-in jugular vein. You can't measure it in your arm or anything like that, blood samples. They're just too dilute. And another very important fact was that actually medical students were the subject for this. Medical students in, I'm not sure if it's Denmark or Sweden, were given CGRP IV, and in migraineurs especially, it was found that CGRP induced a migraine. So, once you know CGRP is important, you want to try and get your drug. So, just like I said at the beginning, there was a high throughput screen, and there was an identification of a dipeptide-like compound that wasn't very special, but chemists that were associated with Boehringer at the time were able to use their knowledge. They'd been working with peptides for some years to get a potent CGRP antagonist. And this was this drug, BIBM 4096BS, that is still the typical experimental drug we used in animal studies today. And as you can see, it wasn't very small. It had a molecular weight of about 1,000, and it has to be injected. It's not orally available. But they had a drug. Oh, I don't know what's happened with this. But I think there's bits here, we've got the bits. So they used this drug, BIBM 4096 BS. They used it in a typical binding assay. And what they showed was that if you take the drug, here's the drug with CGRP, if you take the drug, it could block out the effects of CGRP in terms of binding. So it bound the receptor, and here, go back up, and here, because we knew cyclic AMP was involved in the signaling, when BIBM was there, it's this drug, then things, we moved everything to the right. So it took more of the CGRP when you've got your antagonist there to increase cyclic AMP. Let's see what goes on. I think these titles might all be mucked up here, but never mind. So, got a drug, it works. Where can we go next? We want to get to man. Experiments were done in rats, and it seemed to work in rats, but people knew by then the receptor for human in rats was a little bit different, so they wanted to go somewhere else. And they used a model that we actually devised, and they carried out the experiment in marmosets. And what they did was anesthetize the marmoset, measure blood flow in the cheek of the marmoset, using a laser scanner or a laser Doppler blood flow meter, which we've got here in Pécs in Hungary. And when it was, when you electrically stimulated the trigeminal nerve, you got increased blood flow in the cheek, and that typically was blocked by the drug. So it's blocked by BIBM. And here shows that as the dose went up, you could inhibit the laser Doppler blood flow response. That was enough to get the clearance to test the drug in man, which was a huge step forward. Now, this is a bit of a complicated graph. I'm pleased this has got its title on. So this is one which it was some of the first experiments in humans. So this is the N number in humans, and different doses had different N numbers. And a whole range of doses was tried, and typically in migraine, you look to see if there's a better response after 2 hours. And that's what was found. And what this more simple version has done is just taken the total number of placebo drugs that were tested, and there's also always a placebo effect in migraine clinical trials because people feel they're looked after. But then all the people who had any dose of the drug is shown here, and really there's a highly significant difference. So that was published in 2004, but the only way to give this drug was by injection, not ideal. You always look for side effects, and at this point a lot of drugs fall out of the system because of side effects. What we realized right from the start was that this antagonist had very few side effects, and they weren't bad side effects. And the thing that I was very worried about happening, because it's a vasodilator, I thought there would be lots of cardiovascular side effects, and there just weren't. So you could give this drug at doses which could cure migraine without blocking CGRP that may be involved in maintaining a low blood pressure. And it seems to be that CGRP is not released enough in high enough quantities to influence in blood pressure or blood flow in most migraineurs. So that immediately gives it a bit of an advantage over the triptans. But really, we wanted an orally active drug. So the next thing was to try and develop an orally active drug. In doing that, they managed to develop some antagonists that had liver side effects. So that was a big problem. So the question was, how do you get over this hurdle? At the time, people were studying not only receptor antagonists, but for others, especially TNF, antibodies. Now, antibodies, whether they block the receptor, as shown here, or the CGRP, will act just like a receptor antagonist and block the signal of transduction and block the effect. So, people had already shown that antibodies to tumor necrosis factor and inflammatory cytokine were really beneficial in arthritic patients. And even in a very small clinical trial there, it was immediately obvious that antibodies worked. And I can't remember which one, but one of these people was one of the first in a cohort of 9 to 18 eventually, who took anti-TNF antibody. She was in a wheelchair. I think it's the lady with the darker hair. She was in a wheelchair, and after the drug, she could walk again. So this was held as a miracle at the time and showed antibodies were good drugs. And the more people took TNF antibodies, the more they realized they were brilliant drugs. And that seemed to be a way forward. So what's the difference between a typical pharmacological small blocking agent, antagonist, and something that's bigger? Well, a small drug, such as aspirin, is really small. And it's really not that complex. A larger drug, and I've got here human growth hormone, is larger and is more complex. A large biologic, and this is a typical sort of structure you might get for an antibody for TNF, is huge by comparison and really, really complex. So, should we go for antibodies because antagonists were causing us a few problems in CGRP? At the time, there was lots of debates, and this is a picture from one of our reviews, because people were saying, where will CGRP antibodies be able to get to? Antibodies aren't very well known for getting across the blood-brain barrier, and it's possible where most of the nerves are and the pain, etc., you might need a CGRP antagonist that can freely cross the blood-brain barrier. So antibodies and antagonists, they both be in the periphery, but only certain CGRP antagonists will be able to get centrally. So there's a huge debate saying, will CGRP antibodies ever work? Now, this is a bit of a complicated slide, but it shows that antibodies do work. So we've got antibodies to CGRP here, and we've got the pharmaceutical company that's developed them, and there's 4 of them. They've been tried on different types of patient population, and there's only one that targets the CGRP receptor. The others all basically mop up CGRP, whatever form it is. Now, antibodies have to be, they're not the oral wonder drug, they have to be given by injection, but the point that was realized quite early with antibodies to TNF, and is the same here, is that they actually have a long half-life, and and you really don't have to give them too often. And patients can cope with this quite well. Might be a few days before they have their sort of best results. So antibodies work, and this led to this huge debate of where antibodies actually access the central nervous system. Do they cross the blood-brain barrier? And it's suggested now that they can, more than was previously thought about. And in some of the lectures that we went to at the pain conference we've just been at in Budapest, people were debating how this was actually done. And as I said, there have been more side effects related to the cardiovascular system reported now, but not many. So this is an internet site, Migraine Buddies, and this is a much easier sort of indication of these drugs. And most of them are antibodies. These four are antibodies. But now, finally, we've got some CGRP antagonists coming online. They're being used more and more in the UK and in America and in Europe and in Asia. And Gabor said that one of the CGRP antagonists is also now available in Hungary. There's always a problem, and the problem is they're a little bit expensive. So the way it's dealt with in the UK, where we've got our health system, is that you have to have pretty bad migraine and have to have tried a lot of other drugs before you can access this treatment, unless you're rich, of course. So the timeline was, it was discovered. That's a positive, I think you'll find. People like me were looking at the basic biology and realized it was really significant in that it's a potent vasodilator. It was then realized it was linked to migraine. There was a disaster area when we were trying to learn more about the receptors. I told you it took you some time. What I didn't say is that somebody got the receptor nomenclature wrong completely, the chemistry wrong, on 2 occasions. So it took a long time to get that right, and that held the field back. Then it was realized that what the receptors were, and then that BIBN compound benefited migraine, and then that you have oral antagonists, and there are lots of clinical trials going on with lots of companies. Then you had this liver problem, which held everything back. Then it was realized that an antibody benefits migraine, and then finally it was improved. And now this year, and at the meeting especially, we were hearing about more and more different situations where we can use this drug with benefit. There's also some cautions with respect to its cardiovascular side effects, and they're becoming a little bit more common. So just to summarize, CGRP is known to be released in migraine. We've got the new drugs. They certainly have been effective in all sorts of clinical trials. And you really know they are effective when you have lots of different companies involved and all their drugs seem to be effective. As you test them in more and more, or as they're given to more and more people, you get real life data. One of the problems has been constipation, and they're not now advised to people who get really bad constipation. But Peter Goadsby will say, oh, just give them prunes and everything will sort itself out. So that's his view on the constipation side of it. What does it mean in terms of endogenous CGRP in the cardiovascular system? It probably means there's not enough in us to cause cardiovascular changes when the CGRP is blocked. When I wrote this, I read an abstract from a meeting, and somebody was saying there's one of them, it's the antibody to the CGRP receptor, produces more cardiovascular CGRP events. No, people don't agree with that. Other people who've done similar work don't agree with that. So without doubt, this is a success story in drug discovery. But I just wanted to come back. I've been involved in this all the way through, but I just wanted to come back, turn the concept around, just for my last few slides. If I've got time, 10 minutes, is that right? Or 5 minutes, hopefully. And we've asked the question, will CGRP agonists be beneficial in cardiovascular disease? So there's not enough in us to greatly influence our blood pressure. It's a bit different to the situation with nitric oxide and prostacyclin. But if we add CGRP, could that benefit us? And the first thing we did was carry out experiments in wild-type and alpha CGRP knockout mice. We used a typical cardiovascular model. That's where you have little pumps and you inject mini pumps into the, implant mini pumps into the mice, put in angiotensin II, and you get a high blood pressure. And what we found in our knockout mice was that this blood pressure went even higher. And this was associated with a build-up of fibrosis. This is around the aorta. So this suggests that CGRP, at least in the mouse, is protective. So that was our starting point some years ago. We thought, well, let's try and find somebody who will make a CGRP antagonist that we can work with. The peptide is very difficult to work with because it's a peptide and it gets broken down, etc. So at the same time, there was a USA company suggesting that you could somehow put a gel depot of CGRP in, and that could be good for heart failure. They seem to have disappeared, this company. But our link was with Novo Nordisk, who make stabilized forms of insulin. And they made, for us, a stabilized form of CGRP. It wasn't really for us. They were hoping it might benefit diabetes. That didn't really work out. But what we've ended up here, this is the CGRP peptide, and this is their stabilized ligand. And they've gone from a half-life of 7 to 12 minutes to 7 to 10 hours. So they really increased this half-life. And we were able to use it. The first thing people said to us was, we got funding, thank goodness, to do the experiments, but people said, oh no, this is going to be terrible. You're going to give all your mice migraines. How do you tell if a mouse has got migraine? The only thing we could think of, somebody, Andy Russo in the States, does this, is use a light aversion assay. So we put our mice in a little box, and there's a little hole here, and they can go into the dark. I think you've probably got that here as well, haven't you? Yeah. So we tested what our mice did. Normally, they go from one side to the other, and they sort of spend about 30% of the time in the light, and they did this with our drug vehicle. Didn't seem to make much difference with the dose of CGRP we used, but this was our positive control, glycerol trinitrate, and when they had this, they wanted, they find it quite painful, and they wanted to be in the dark. And glycerol trinitrate can induce migraines. So that was our test to show we weren't completely mad. We then took these, we were advanced by this point to do experiments by telemetry. This is a baseline blood pressure, and this is where the mouse has got a sensor in it, and it will transmit to a reading system beneath the cage. Angiotensin II gave all our mice hypertension, and then we had two different groups. One group which received vehicle, so that was basically untreated and maintained a high blood pressure. And this was what we got with our CGRP analog. We had to inject it, so we got a decrease in blood pressure. But it seemed to sort of stay low from what we could see. So we thought this is interesting in terms of blood pressure, but loads of drugs for blood pressure, not many for heart failure. What happens in heart failure? So we carried on, did experiments with an abdominal aortic constriction model where you constrict the aorta, and this puts pressure on the heart. It's an overload model. And we looked at heart function just to see what happened, and histology. Now the next slide is a little bit confusing and complex. I just want to show you one bit, and that was that our stabilized analog, when it was given, seemed to revert the mouse, even though the mouse had heart failure, it reverted the mouse back to a normal phenotype. So this is the mouse with heart failure. It's got a reduction in ejection volume. There's an increase in the septum wall thickness, and there's generally there's another difference at the end here with left ventricle length, which means that the heart is normalized. So CGRP treatment seems to normalize the heart. So far, we've gone on now, and our latest studies, which aren't published, are in a model of myocardial infarction. But so far, we've shown 4 models that shows a therapeutic potential for the injectable CGRP analog. It seems to protect against the onset of the hypertension and the cardiovascular remodeling. It's effective over 5 months in that AAC, 5 weeks in that AAC model. In our myocardial infarction model, we've done something a little bit more exciting. We only gave it from 1 day to 4 days, and then we followed the model for another 3 weeks, and it seemed to, the protection we had seemed to be maintained. And we don't seem to have, in the mouse at least, obvious side effects. So the person at Novo Nordisk that we worked with, she's now got her own company, and she actually owns this company. When she left Novo Nordisk, she was given this compound, which was quite a nice leaving present, I think. They weren't interested in it because it didn't do anything in diabetes. And she's now shown, she's doing studies in the pig where the results look promising. So to conclude everything that I've been talking about, CGRP blockers have changed the treatment paradigms in migraine. In addition, we think that stabilized agonists from the CGRP family can be beneficial in cardiovascular disease, especially heart failure, where there really aren't many drugs, as I'm sure you know. And at the moment, we think we can go ahead using an injectable stabilized CGRP agonist, just because you only need to give it for a short period after the heart failure. So that brings me to the end of this lecture. I just wanted to sort of point out the people who did the work. The early work on CGRP and knockout mice was done by Sarah Smiley. Aisha Abdul did the abdominal constriction model, and more recently, Fully Arginine has also been working on this project, but lots of other people as well. And Gabor did a sort of a project that sort of went a different route that involved TRP receptors as well, but also involved CGRP to a bit when he was with us. And Erica, many moons ago, when she was working with us, she was not very far from CGRP as well. So that's it. I think I'll leave it there. Great.
Thank you, Sue. I think it was an absolutely comprehensive lecture about CGRP. Just this is typical during the drug development. CGRP antagonists and antibodies beneficial in treatment of migraine, but possibly very serious side effects, because CGRP is a well-known vasodilator. It's a very important essential peptide in the cardiovascular system. And now this is open for the question. We are waiting for some notes and questions, especially from the students. The Hungarian National Scientist Academy. Or my colleagues, maybe Balázs can ask. Please come here. I can give you the microphone.
Yes, thank you. Yeah. So it was a lovely lecture, thank you very much.
Thank you.
And I was interested that in the early days, in the first approved anti-migraine drugs, like the triptans and so on, it wasn't necessary to prove any pain alleviation in rodent models to get clearance for phase 1 clinical trial, but some other sorts of physiological changes like the CEMP levels and blood flow were measured rather.
Yes, so in the early days with the triptans, most of the experiments were done in what some people might think is a little bit old-fashioned these days. They were done on bioassay, and then it seemed, if anything, to be easier to get human tissue, especially sort of studying what happened in the coronary artery, because they wanted something that was quite weak in contracting the coronary artery, but also effective in the cerebral vasculature. So they did a lot of their experiments in humans, but there was a very important model in the rat where the trigeminal nerve was electrically stimulated, and that caused neurogenic inflammation in the meninges, which could be measured. And also, in the anaesthetized rat, blood flow could be measured as well. So experiments in the anaesthetized rat especially were really important for the triptans, and similar experiments were carried out with when looking for what roles CGRP may had and what roles CGRP antagonists had. In terms of trying to work out which compounds were CGRP receptor blockers, there you could do an awful lot of work in culture, in cells. So typically in this area, people have done a lot of work in culture. They've utilized preclinical models, and at the time it was mainly the rat. It's still, I think, found quite difficult to use for some of the neuroscience experiments that involve the CNS, the mouse. But also, everybody tried to use human tissue when they could. When they carried out the first experiments with a migraine, that result about the 2 hours, how many patients had relief after 2 hours, that seems to be the mainstay. And we heard a lot about that this week. That's still the main test. Get the drug into humans, see if there's benefit after 2 hours compared with placebo, and that will tell you. Yeah.
And was this performed similarly to check if there are no real adverse effects of CGRP agonists for the treatment of heart failure?
Yes, so nobody's got that far with the agonist for the treatment of heart failure because we haven't got any further than the pig. What we're finding, which is quite interesting, is that, of course, everybody wants better drugs for heart failure, but getting to the stage we are now, there's been an awful lot of failures. So the people who pay for these experiments, the first clinical trials in humans, they really want everything sort of practically shown before you do the experiment. So every time you get near to any sort of funding for a clinical trial, somebody will ask you to do a whole load more experiments than some other species. And of course, as I'm sure you probably know, that doing experiments in the more human-like species is very, very difficult to primates and marmosets, etc. It's absolutely not easy, and it's getting more and more difficult. And people like us would like to avoid it and just take the risk, but other people have different opinions. Yeah.
Thank you very much.
Yeah.
Thank you, Balazs. Any other question from the audience, young audience? Yes, please come here, because otherwise we can't hear your question. Angelica?
Thank you. First of all, thank you very much for your very interesting lecture. And I was wondering that is it known how is it how the CGRP is related to the signal transduction related to cardiovascular remodeling? So, do we know or are there experiments currently discovering?
Yes, that's a good question. So, what seems to be the situation is that there may not be much endogenous CGRP, but there's an awful lot of CGRP receptors around in the heart, be it on the cardiomyocytes, be it on the vascular cells, and because CGRP increases cyclic AMP, also that can act in a protective way to stop some of the inflammation. So in the latest experiments we've done, straight after, this is in the mouse, straight after myocardial infarction, you find you get edema formation and neutrophil accumulation, and even at that stage it looks as though CGRP can be protected. So, and now I realise that there's some evidence that where that early inflammation is where the fibrosis sort of comes later on. And I'm now told that clinicians, suddenly it's becoming very trendy to try and work out what happens in that very, very early stage in humans during that time when there's the edema and the swelling, really very soon after myocardial infarction. So I think it's all starting to make sense. And if you can just get CGRP into humans early enough, there might be. But everybody's looking for drugs that you could treat heart attacks by just getting them in really early. There may be, yes, it may be a very good candidate to help. Yeah.
Thank you very much. And I have one more question. Are there any changes with age regarding CGRP levels? It only came to my mind because migraine is mostly related to younger people, while heart failure is usually present in the older days. So maybe is there any connection with that?
Yes. So there's evidence that as you get older, CGRP levels go down, but this is very much debated because, of course, CGRP levels go down, but on the other hand, pain-related phenomena all increase. And so I think that's a very unclear sort of area, but it certainly makes sense to me that as you get older, CGRP levels go down. Another point is that if you have a diabetic neuropathy, there you lose your nerves, but you've still got huge pain. So that's another. We don't know that much about this area, which for me makes it quite an exciting area where we've still got a lot to learn. Yeah.
Amazing. Thank you very much again.
Yeah. Thanks.
Thank you, Angelica. Any other question? Just, Sue, I think we had a common grant more than 10 years ago. I worked with CGRP knockout mice on a scleroderma model.
Yes.
And autoimmune diseases, scleroderma or Raynaud. Just in this case, the peripheral vasculature is very important to maintain the blood flow. And I think in this case, inhibition of the CGRP could be dangerous. Do you know something about it?
Well, to be honest, it was only at this conference I've seen any warnings. So CGRP antagonists and antibodies have been given to loads of people now with benefits in migraine and minimal or no side effects. But just in one person's talk, there's a suggestion that if you know somebody needs a lot of needs to go through a wound healing phenomenon, they might have just had some sort of accident of some sort, or perhaps even a sports injury, that you should think a little bit about whether you give them CGRP antagonists or antibodies. And there was another suggestion from this same person. I hadn't seen such a detailed list before, that if you have somebody, and of course you do get younger females especially who suffers from Raynaud's disease, it's that phenomenon when you're outside and all the tips of your fingers go very cold and painful, or toes, etc. In that situation, it may not be a good idea to block one of your ways to vasodilate. But until now, I've not seen any limitations. The only limitation I've seen is that if you suffer from high blood pressure, then it's suggested, and there's a warning on the packet that you don't have the CGRP receptor antibody, just that one.
Yes, but I still think this is a real danger.
Yes,
We will
yes,
see next time.
yes, yes, yes.
Thank you. Any other question? No? Okay, if not, now I would like to give you a certificate. Our program would like to accept you as the international mentor of Pécs program, National Scientist Academy. I would like to give you this certificate.
So I've got to stay here, I think.
Yes.
So
And
thank you
I
very
think-
much indeed.
I put it here. Congratulations.
Thank you.
And please, get an official photo with you.
Yes, thank you.
Could you take a photo together?
Thank you.
Thank you very much again. And
That's lovely. Thank you very much. And it's a real honour, and I hopefully there's a chance to chat or whatever. Yes.
This acceptance, sorry. This acceptance means your regular visit here, and hopefully we can continue our fruitful collaboration in the field of neuropeptides, CGRP, substance P, or TRP channels.
Great.
Thanks again.
Thank you very much indeed. Thank you. And thank you, everybody. I realise for the students it's not an ideal weekend for me to visit, but thank you.